Hello, everyone. My name is Jen Yost. Thank you for coming. I'm in my jammies. I hope that you are too. Today, I'm going to be talking about a large-scale imaging project that I run in California, the California Phenology TCN. This is an NSF-funded ADBC project, which is focused on imaging specimens from California. To put that in a little bit of context, our work is focused in California, which is a biodiversity hotspot. It's also one of the world's 5 Mediterranean climates, and it harbors a huge amount of diversity in the United States. Within the state, we have 3 different floristic provinces represented, over 5,000 native plant species, 30% of which are endemic in California. And that is despite the fact that 3/4 of the original habitat in the state has been lost and developed due to this population of over 40 million, and it's likely that 40 million more people are on the way. Half of the food of the United States is grown in California, and yet it is still one of the most biodiverse places outside of the tropics. So we have many unique and rare species within the state, and at the same time, we are in a period of climate chaos, with California getting drier, rainfall being more sporadic, and our fire season longer and more devastating. So that begs the question, how will our flora respond? This is what's motivated our project. We're interested in which species are in the most danger of phenological shifts. What are the impacts of those shifts, both in terms of agricultural plants, pollinators, pathogens, and pests? Which habitats and vegetation types are most phenologically sensitive? And what is the effect of precipitation and temperature on a plant's phenology? And so this led to the creation of this digitization project. And our goal is to build a data set that answers these questions. We're doing that by building a historical record of flowering time using herbarium specimens housed at California Herbaria. Initially on our project, we had 22 funded partners, although now we've brought on about 6 more. Our project is led by myself at Cal Poly, and KD Pearson is our project manager, Jason Alexander and Brent Mishler at UC Berkeley, Ed Gilbert at ASU. Initially, at the start of this project, we had these 22 institutions that spanned the state, but we've been able to leverage the funding from this project and really KD's time and effort in training people to include many more institutions. So now we have over 45 institutions that are receiving training and support as part of this project. The goal was to create images of 1 million specimens. Each of those specimens would have a transcribed label, a georeference, and a phenological score associated with them. Now, we couldn't image everything, and so we have 22 target families. These families represent the oldest records within California, so we can extend the historical flowering time back as far as possible. It also includes the most diverse families within the state, also the families with the highest rates of endemism, and also the most threatened families. In addition to those, the National Phenology Network has a network of citizen scientists that monitor flowering times on living specimens, and we included 250 taxa to match the species that they monitor. All of the data that we produce is now aggregated into a new California SYMBIOTA portal called the CCH2. This is a publicly accessible portal where you can search all of our records. Currently, we have 54 collections, over 3 million records, about half of which are georeferenced, 23% are imaged, and 90% are transcribed. This is where we're housing the images produced on this project, and so you can log on there and look at all your favorite specimens. One problem that we encountered when we were developing this project is that there are not standards for phenological traits from specimens. And so part of this work has been to establish a framework for which trait-based data can be shared in Darwin Core archives. In 2016, a group of phenology experts got together and created a draft protocol for how this would look. And essentially that breaks a phenological score on a specimen down into 3 different orders. The first and broadest category is just answering the question, are reproductive structures present on the specimen? Yes, no, or not scoreable. And for many species, this will be all that you're able to answer. However, for many other species, you can probably answer second order questions, which is, are unopened flowers present, are open flowers present, and are fruits present? And then we tried to plan for the future in which some researchers might want a finer look at where in the phenological trajectory a plant is. And so a third order question could be something like counts or percentages for the number of buds, flowers, or fruits on a specimen. We've tried to keep our vocabulary as much as possible consistent and mapped to the plant phenology ontology so that we are not reinventing words. Part of this project has also led to many new developments within SYMBIOTA. And so in our portal, in addition to the occurrence editor, which is what you're seeing here on the screen, there's a new tab, the traits tab. And we've designed this tab so that it could accommodate any traits that you might collect on a specimen into the future. For our project, we've defined a trait as the angiosperm phenological traits, and you can click on that tab and then see the scoring or the phenological status of that specimen. You can also apply a phenological status to that specimen from this traits tab. Now, this is available in all SYMBIOTA portals, so if it hasn't been activated in yours, then you should get in touch with your portal manager. Along with this, there are 2 new features within the editor control panel. This is the ability to create trait data, either from looking at an image or from mining the text that's captured in some of the label fields. Here is an example of what the image scoring editor looks like. You're presented with an image and you have the trait of interest, and then you can apply a phenological score to this specimen and move on to the next image. And you can filter that by a geographic region or by a taxonomic status. This is an example of the trait mining feature for verbatim text. So many times phenological data is captured somewhere on the label, but it's just been done so in a non-standardized way. And so what you're able to do here is you can apply this trait, which is our phenology, and then you can search a various field of your database, in this case, reproductive condition. And then you'll see every unique value that exists in that field. And you can select all of the values that apply to having flowers, and you can apply a standardized scoring to them all at the same time. This is how the data are captured within SYMBIOTA, but those data then are shared via a Darwin Core measurement or fact file. And this gives you a bunch of metadata about the variables that you're providing measurements for. And now within our SYMBIOTA portal, when you download occurrence records, you have the option of including trait attributes with that output file. Now this will download as a separate Excel file, and you have to do a little bit of work in order to merge that file with the occurrence record. As of right now, our project is just finished year 2 of 4. And we've made various progress on imaging, transcription, and georeferencing. Everything is going well. But what I'd like to draw your attention to is that using those tools that I just described, we were able to phenologically score over 1.3 million specimens very quickly. And so this really took advantage of the fact that these data were being recorded somewhere on the specimens, and being able to just translate that into a standardized view now makes available over 1 million records that can be used for phenological research. Another aspect of our project is that we involve the community in Notes from Nature expeditions. So what a Notes from Nature expedition is you can go to the site, you can click on various projects. I've highlighted the Capturing California's Flowers project. We have a number of expeditions taking place where various herbaria upload their images, and what you're able to do then is help us transcribe records that are not digitized yet. There's great tutorials and help menus for how to extract meaningful information from the label. Now, what I'm showing you here are transcriptions per day over the last few months. And it's interesting to note that right here is when California's shelter-at-home order took place. And so this is an argument for how important digital records are, and that they enable people to engage with these collections from the comfort of their own home. And I hope that this uptick continues. Another aspect of our project is teaching undergraduate students how to use biodiversity data in research. And so last spring, we launched this class for the first time. Our Cal Poly was a completely virtual quarter. And so we ran this class over Zoom. It was actually incredibly successful, and it worked just fine in the online forum. We were able to usher 18 students through the process of picking a species, using the images generated in the CCH2 for them to georeference those specimens, apply phenological scorings to those specimens. And then a couple weeks in, they downloaded all that data, including PRISM climate data, and then they were able to ask and answer meaningful questions about their species and produce a scientific poster at the end of it. So if you'd like to hear more about that, KD Pearson is giving a whole talk on that, which is abstract number 176. The last thing I'd like to draw your attention to is our website, capturingcaliforniasflowers.org. On this website, we are accumulating many resources, whether it be research papers regarding phenology from specimens, but also all of the course materials from that class are available on this website, as are all of our protocols and recommendations for the digitization process. My contact information is there, as well as KD Pearson's, and you can follow us on Twitter. I'm happy to take any questions, and thank you all for listening in such an odd format.